Viagra, also known as sildenafil, is a drug commonly prescribed for erectile dysfunction as well as pulmonary arterial hypertension. Erectile dysfunction is defined as the inability to get or keep an erection firm enough to have sexual intercourse. Viagra could be separated into three parts, including the pyrazole primidine group, an ethoxyphenyl group, and a methylpyrazine group. These key features of Viagra are essential for binding to the PDE5 and give the antagonistic effect. The propyl group is associated with favorable selectivity of binding to PDE5 over PDE6. As you can see from the structure, Viagra adheres to Lipinski's rule of 5. It contains 8 hydrogen bond acceptors, shown in red, less than the allowable 10, 1 hydrogen bond donor, highlighted in blue, less than the allowable 5. It has a molecular weight of 474.58, less than 500, and a log P of 1.5, less than 5. All of these factors allow the drug to be orally available. But before we talk about how Viagra works, we must first explore the mechanism behind a normal erection. Stimulation, usually of a sexual nature, causes the parasympathetic nervous system to release nitric oxide from the nerves. Nitric oxide diffuses into the carbonosal smooth muscles and increases the intracellular levels of cyclic GMP and cyclic AMP. These cyclic AMP and cyclic GMP activate the protein kinases, which phosphorylate certain ion channels and lead to the opening of potassium channels, as well as the inhibition of voltage-gated calcium channels, leading to a decrease in calcium. The relaxation of the smooth muscles, as well as the dilation of arteries and arterioles, allows the lacunar spaces to fill with blood and the occurrence of an erection. Now, onto the biological basis of the condition. The causes of erectile dysfunction include, but are not limited to, obesity, heart disease and atherosclerosis, diabetes, depression, kidney disease, and hypertension. Now on to the discovery and development of Viagra. Through the discovery and development process, we can see how the focus of treatment for sildenafil changed from being an angina treatment to overcoming erectile dysfunction, which gave rise to the drug called Viagra. In 1986, Pfizer scientists formed a research team to develop a selective phosphodiesterase type 5 inhibitor. Whilst looking for anti-angina drugs, research discovered that Zapranist, the molecule shown below, had some effects that they were looking for, including vasodilation. This was due to its inhibition of a phosphodiesterase type 5 enzyme. 3 years later, a drug called sildenafil was developed for further profiling and entered clinical development for cardiovascular indications such as angina. A series of clinical trials in 1993 showed that sildenafil gave inconclusive results in the treatment of angina due to its short half-life and interaction with nitrates. Therefore, the decision to undertake pilot studies with sildenafil in erectile dysfunction was supported by the observation that penile erections were a common side effect in the multiple-dose sildenafil phase 1 study. In 1994, 20 clinical trials were conducted, which involved over 4,500 people, and this proved the safety and efficacy of the drug. Subsequently, sildenafil was patented in 1996 and was later accepted for prescription use in most countries of the world by 1998. The development of an oral formulation was perceived to be a breakthrough in the pharmaceutical world, as advances were only starting to be made in recognition and treatment of erectile dysfunction during the 1980s. Prior to then, many clinicians considered the condition to be a trivial issue or attributable to psychological causes. Now onto the synthesis of Viagra. The first synthesis of sildenafil involved 11 steps. Some of the key steps will be shown below. The first step was the reaction of a diketoester and hydrazine to give a pyrazole ring. The second step involved the methylation with hot dimethyl sulfate. The third step was the base hydrolysis to give a carboxylic acid, which would then undergo nitration with fumic nitric acid. The carboxamide is also formed with refluxing of thionyl chloride or ammonium hydroxide. The compound is then acylated with 2-ethoxybenzyl chloride. After that, cyclization occurs under basic conditions to form a lactam ring, which is then sulfonated with chlorosulfonic acid at the 5 position to give a chlorosulfonal derivative. The last step is condensation with 1-methylpyrazine, and the final yield of Viagra is 88%. As this reaction requires many stages of synthesis and purification, it was originally not a very efficient process to satisfy the demand. As a result, the drug company developed a more efficient method by changing the initial products to these more complex compounds so as to reduce the steps of purification and the processes that are required. The production method now is not just more efficient, but also produces far less waste as well. The role of phosphodiesterase type 5 is regulating the concentration of cyclic GMP and the relaxation of smooth muscle. Cyclic GMP binds to the allosteric site of phosphodiesterase type 5 and is released after finishing catalysis. Viagra is used to inhibit phosphodiesterase type 5 in the penile corpus cavernosum. It blocks the catalytic site and prevents the catalysis to increase the concentration of cyclic GMP inside a smooth muscle. This enhances the rate of duration of erection. As mentioned above, cyclic GMP and Viagra are both substrates of phosphodiesterase type 5. By comparing their structure, you may notice that there are some similarities. Therefore, this part of the molecule is the pharmacophore for binding of PDE5. The catalytic site is composed of 4 subsets, including the metal binding site, the core pocket, the hydrophobic pocket, and the lid region. At the metal binding site, there are 2 metal ions. These are zinc and magnesium. However, there is no known interaction between cyclic GMP and PDE5 inhibitor. Pyrazole pyrimidone group of sulfonylurea has an interaction in the core pocket. The core pocket also forms hydrogen bonds with pyrimidine, nitrogen, and the carboxyl group of the ring. There is also face-to-face pi-pi stacking with the conjugated aromatic rings. The hydrophobic pocket forms hydrophobic interactions with the ethoxyphenyl group of the sildenafil, and for the lid region, the methylpyrazine group of the sildenafil forms hydrophobic interactions with the binding site as well. However, this part is responsible for the breaking down of cyclic GMP into GMP, and therefore this is the part of the molecule that has the blocking effect towards the active site. There are a number of side effects due to the enzyme phosphodiesterase type 5 being present in the vascular smooth muscles, such as in blood vessels and the digestive tract. This leads to a number of side effects, the basis of which is due to the effect of systemic vasodilation. On-target effects include flushing, headaches, nasal congestion, dizziness, and hypertension. Relaxation of the lower esophageal tract can also cause gastrointestinal side effects, such as dyspepsia and nausea and vomiting. Another rare but sometimes serious side effect is priapism, which is a painful, prolonged erection lasting longer than 6 hours. Viagra is also a weak inhibitor of the enzyme phosphodiesterase type 6, showing roughly 1/10 of the selectivity of type 5. Because it is present in the retina, an off-target side effect can be things such as visual disturbances, changes of colour perception, such as blue-tinted vision, blurry vision, sensitivity to light, and increased intraocular pressure. The most common side effects include nausea and headaches, only affect about 1 in 10 people. However, generally, side effect incidence is fairly low and less than 1% of users suffer from serious side effects. Since its introduction in 1998, over 20 million men have used or are currently using Viagra to treat their erectile dysfunction, showing just how significant of a breakthrough it was in the pharmaceutical world. Thanks for watching.